Good morning to everyone. I respect personal head um, and my dear colleagues and dear students. Good morning to all. We are happy to. It's our honor, and I'm happy to have Dr. V. Mohan Rai sir, who is my teacher in GKVK Bangalore. Sir worked on many areas, including temperature index and response, and sir worked on rice. Now I came to know that sir is working on potato, but he's a well-known stress physiologist and also a good teacher, very good teacher. He handled classes. I think you are seeing how he interacted. No, it's a rather I say GK said where students are not allowed to move a slide unless they understand. It is not they ask at the end. Every slide you move, there'll be questions from all our sides. So that kind of I think we need to have that kind of interaction where you have to have questions from all corners. Also, I expect our students also to have that kind of interaction, to have ask questions, and it's our no privilege. And sir, happy to listen to you, sir. That's you, sir. Please, thank you.
Thank you, Bhumi, for the very nice introduction. And I don't know whether I have to say the good morning or good afternoon because it's already 12 o'clock now. Anyway, I work on different areas, and this is one of the areas which we started working recently. And I thought it would be more appropriate to talk something else rather than what we have been doing in the field of physiology, because we know that we have been talking about many kinds of stresses, mainly the aerobatic stresses like the drought and the salinity or the temperature, which we have been hearing for a very long time. That so then I thought telling the same thing has no meaning because most of the things you know about. So I chose this topic mainly because of the fact that we had one NSF funded projects which we completed recently. So I thought I would be I would share my whatever the research outcome, so that would be more appropriate. Today I'll be talking on the and before I start my the talk, I don't know how much justice that I can give it to this August body. Okay. I'll try my best to convey whatever we have done and whatever the literature informations are available with respect to the tuberation in potato. Now, the title that I've given is the tuberation in potato, the physiological and molecular mechanisms. Right? We know that the potato is one of the the important vegetable crops, right? So where it is mainly been consumed for the starch, mainly it contains about 20% roughly around 20% of the starch and about 2% of the protein and less than 1% of the fat and things like that. And if you go back to this potato, it has originated from Andean region of the South America, like in Bolivia, then Colombia and the Peru region, and belongs to the Solanaceae. That. And having said that, it belongs to the Solanaceae. Probably I don't know how many of you worked on potato. That it's a very difficult crop to work with because it has lot of the diseases. So the raising the crop itself is a Herculean task, unlike the other Solanaceae member, where you can easily they grow the crop. The potato is a very, very difficult crop because of a lot of the disease incidents, right? So that is one thing, and it's a cool season crop, and therefore it has to be a cool season crop, and it's generally been grown in Rabi season where the conditions are very cool, the temperatures are cooler, and the photo period is shorter the photo period conditions. That's the thing that is required for the proper tuberations. We are interested in potato for the tubers, not for flowers, and it's a short day plant, especially short day for the tuberation, and it requires a temperature of around 18 to 20 degrees centigrade for the proper tuberation, and therefore it cannot be generally grown in Kharif season. Rather, it's been cultivated during the Kharif season where the conditions are very favorable. As far as the potato cultivation is concerned in Indian scenario, the potato is being cultivated and consumed to a large extent in in northern India, where the conditions, the climatic conditions or the environmental conditions are very favorable for the potato cultivations, and therefore, you could see that the most of these the potato is being rather been confined to the North Indian conditions mainly like Shimla, Punjab, and other parts of Uttar Pradesh, where the conditions are good. That, and more importantly, what I would like to also to share with you is that the South Indian conditions is not really favorable for the potato consumptions. Probably that could be one of the reasons why the potato consumption, like unlike the people from North India, where almost every day they consume the potato. That, but we people from South, I don't think when I go to market, I just bring the potato maybe once or twice a month. So we don't consume the potato on a daily basis. Rather, it will be like once, probably once in a week, or maybe 2-3 times in a month. So both the cultivation as well as the consumption of potato is rather been low in South India compared to the northern parts of the country. And more importantly, what I also wanted to share with you is that the potato seed material which we are required to cultivate has to be invariably come from the northern parts of the country, that especially from the Jalandhar and other areas where you have large places where the potato tubers are being produced. And these potato will have to travel all the way from North India to the South India. That, and imagine what will be the kind of the cost involved that. So if you now work out the total cost of the production, nearly 50% of the cost goes for only the transportation. So therefore, the seed material accounts for nearly 50% of the total cost of the cultivation. So it's totally a very huge amount is rather been incurred towards the procurement of the seed material, mainly because of the fact that the South Indian conditions is not really congenial for the production of the seed or the seed multiplication. So in this scenario, what we thought was, can we think of growing this potato, or producing the potato tubers in South India with some controlled conditions? We made an attempt and we were successful even during Kharif season, which I told you that the Kharif season is not congenial for the potato cultivation. But through creation of the congenial conditions, it's possible that even in Kharif conditions, it is possible to produce the potato or the tuberize, to make the potato plant to tuberize them. So that's what one of the outcomes of the project work. That, well, there are a lot of constraints for the potato cultivation. Major constraint is the quality planting material, which is one of the major constraints as far as the potato is concerned. That, and as I told you that 50% of the total cost goes for only the procurement of the seed material. And why is that the South Indian condition is not favorable is because we have a lot of viral load, and it is not it is not possible to produce the virus-free planting material, which is rather been possible in the northern parts of the country. And that's the reason why we are getting the viral-free material exclusively from the northern parts of the country. Now, we made an attempt to grow these plants through the aeroponic conditions, aeroponic technology, where through this the aeroponic technology, it's possible to develop the disease-free planting material and also minimize the cost for the cultivation, because we can totally cut off the amount of money that we spend for the transportation of the material. And another important thing is that it can be cultivated throughout the year. That, okay. So if you have any questions in between, please ask, whatever the kind of the stigma that has been attached with that, you should ask the questions only in the end. I think you should take it out and ask the questions whenever you feel like, and clarify the doubts. Is it okay? Am I going fast, or will you follow? Are you following it? Okay. Okay, fine. So
Actually, the planting seed or
the tuber itself, tuber, depending upon the number of the hybrids you have, that you can make it into number of pieces. Like if you have more number of the hybrids, accordingly you can make it into small pieces, and see that will be serving as a planting material. Okay. And as I told you that the potato tuberation is a very complex kind of things, like how Bhumi also knows that we have been talking about the kind of the drought tolerance. When we talk about the drought tolerance, we always say that it is the highly complex, and developing the drought tolerant crop varieties is extremely difficult. So in a similar way, when you look at the tuberation in potato, the potato tuberation is also regulated by a number of the factors. That we have positive factors which will influence the tuberations. We have negative factors which will inhibit the tuberation. As far as the negative factors are concerned, the long day conditions are not inducing the tuberation, so it inhibits the tuberation. So therefore, whenever you have in air, if you have the longer day conditions, that is generally being prevailed in the Kharif season. So it is not favoring the tuberation that. And more importantly, the temperature. The temperature plays a very, very important role as far as the tuberation is concerned. Whenever the temperatures are higher, it is certainly inhibit the tuberation in potato. So therefore, the temperature and photoperiod are the two major important environmental variables that will really play an important role in terms of inducing the tuberation. So these two factors, high temperature and the long day conditions, are not really favoring or rather inhibiting the tuberation. In addition to these, like hormones, gibberellic acid, we know that we studied as a physiologist, we know the importance of the gibberellic acid. So gibberellic acid has multiple roles. We know that without the gibberellic acid, many of the plant processes are not being possible. But unfortunately, when the gibberellic acid is playing so much of importance in the plant's overall growth and development, when it comes to the tuberation, the level of gibberellic acid has to come down. So we need the lower gibberellic acid content to induce the tuberation. So rather, we require more of the cytokinin, which is now important for the tuberation. And more nitrogen. And again, we know that the nitrogen is required for the proper development of the plants. That's the reason we apply more and more amount of the nitrogenous fertilizers to basically to boost up the vegetative growth. And it doesn't mean that the nitrogen is not required for the potato, but certainly the nitrogen is required in the initial growth. So when the plant is in vegetative phase, certainly we require more amount of the nitrogen. But when the plant enters into the goes into the tuberation period, at that point of time, the level of the nitrogen has to come down. So these are the factors which are acting as a negative regulators, which will rather inhibiting the tuberation. So when you look at the positive factors, it requires low temperature, it requires the short day conditions, it requires instead of GA, the level of GA has to come down. At the same time, the level of cytokinin has to be higher level, and more importantly, the nitrogen content should also be lower. And imagine controlling all these factors and putting together into a basket is extremely difficult task, and that's the reason why the tuberation or the potato cultivation is not really possible in all the seasons, except that in the Rabi season where the temperatures are lower, as well as the short day conditions are prevailing with the lower level. And naturally, at the time of the tuberation, you could see that the GA level goes down. At the same time, the cytokinin level is increased, which will favor the tuberation. Well, these are the conditions. For example, if you now look at the tuberation between the Rabi season and the Kharif season, Rabi season is seems to be very optimum for the tuberation, where you can see that the temperatures are optimum. Again, photoperiod will also be optimum. When you look at the cumulative day temperature and also the cumulative night temperature in Rabi season, oddly, you'll have the temperature around 22 degrees centigrade day temperature in Rabi season, with almost about 16 to 17 degrees temperature prevailing during night period. Right, and even the light durations, if you now look at the light durations, it's about 10 hours of the light period and the remaining 14 hours of the dark period. So therefore, congenial, the conditions are very congenial for the tuberization in the Rabi season, and that's the reason why in most places in South India we prefer to grow the potato only during the Rabi season. However, when you look at the conditions during the Kharif season, so look at this, the temperature. The temperature is, although we get a lot of rains and all those things, still the temperatures are higher, where the temperatures are nearly about 26 degrees centigrade day temperature, and the night temperature will be around 20 degrees centigrade. So, and the photo period is slightly longer. So therefore, these conditions are not really favoring the tuberization in the Kharif season. So one of the challenges in the potato cultivation to produce the potato tubers is to how best we can think of producing the tubers in Kharif season, in spite of having so much of the negative factors prevailing during the Kharif season. So, and of all these factors which I just discussed, the temperature is the one which is playing a very crucial role, which is rather been very, very important for the tuberation is concerned. So this temperature regulation is possible when you grow the potato plants on aeroponic chambers, where it's possible to regulate the temperature both in root zone as well as in the shoot zone conditions. So this is what the snapshot of the aeroponic chambers that we developed and control the temperature in the root zone. That so initially what we done was we developed the aeroponic chambers, and in that aeroponic chambers we started growing the plants. And I told you that to standardize to grow the plant itself, it took almost 1 year. That so initially we struggled a lot to raise the plants, mainly because we have a lot of viral diseases. Controlling this viral disease itself has taken a lot of time, and we were successful. And after about a year, we were successful to grow the plants on the aeroponic chambers. And this is what has been the scenario with respect to the cultivation of potato on the aeroponic chambers, where we have two the areas. One is the inner portion is the root zone, where the plants have been planted in such a way that the shoot system is above the chamber, and the root system is inside the aeroponic chambers. And these aeroponic chambers have been sprayed with the chilled nutrient solutions. You know that being the physiology students, you know the hydroponics and aeroponics, where the aeroponics is where the plants are being grown in suspended air. And these the plant system are not in contact with the soil, they need to be provided with the nutrient solution. So we have standardized the nutrient solution required for potato, and we also standardize how often these nutrient solutions are to be sprayed, and what is the composition of the nutrient solutions required, and all those things. Everything has been standardized. And we also since the temperature has to be regulated, so we used a chiller which chills the nutrient solution, and the chill nutrient solution was sprayed onto the hanging root system. And with that, we we could see that we have regulated the root zone temperature. That if you now look at, do we have a pointer there? No pointer. There is a pointer. I can understand that. Anyway, okay. So if you now look at the temperature regulations in the aeroponic chambers, you see that the red one and the other one is the light green color one. So the green color one is the one which is where the root zone temperature has been maintained almost around 18 degrees centigrade throughout the experimental period. So that means it's possible that the temperature in the root zone can be maintained through spraying the chilled nutrient solutions onto the root system, so that the temperature is being maintained all through the experimental period in the root zone. However, during the daytime, what is happening is, if you've not been able to spray this, we also maintained the another chamber where we have sprayed only the nutrient solutions without any chilling treatment. So therefore, we could see that there is a rise in temperature in the root zone system. Okay. So with that, the rise in temperature, it has affected the tuberations, which I will be showing the data. See, this is what the root system, the hanging root system, we have sprayed the nutrient solution, right? And because of that, the maintenance of the optimum temperature of around 18 degrees centigrade, we were successful in inducing the tuberations, where you can see that the tubers are, these are all the mini tubers. I just wanted to make it very clear that the tubers that we have produced in the aeroponic chambers are the mini tubers, that they are small in size. It will be of about 3 to 4 grams. That's the size of the tubers, which will be rather used as a seed material. Now, again, I also want to tell you that these tubers which have been produced are the virus-free material, which you have checked that they are, because the plants that we started putting on the aeroponic chambers are virus-free, which you have generated on through the tissue culture method. And subsequently, we have planted on the aeroponic chambers, and we also confirmed that these plants are virus-free. And the tubers are obviously been virus-free. And these tubers, once you produce, they will not be directly taken into the farmer's field. Because if you're now taking these the tubers directly to the farmer's field, it will be, the cost is very high. That each tuber, a small tuber of about 4 grams, is costing you around 10 rupees. That's the cost of the production of the mini tubers, which is virus-free. So therefore, you cannot sell these tubers, or you cannot directly take these tubers into the farmer's field. What generally people do in the northern Indian condition is that they actually, like these mini tubers, don't go with an assumption that these tubers can be directly taken to the farmer's field. So it will be multiplied in the net house conditions, where there are no vectors gets into the net house, and this been multiplied for several generations. For about 5 generations, these tubers are multiplied. Like one tuber when you grow and are taken into the 5 generations, at the end of the 5 generations, one tuber gives rise to almost 1,000 tubers. That. So after 5 generations, these tubers are being sold to the farmers, which will be used as a seed material. That. Okay. Now that's what I thought. Also, yeah. It is because of the aeroponic system. That aeroponic system. Otherwise, if you grow these tubers, you plantlets in the field conditions, it will be as big as any other the tubers.
This condition can get more number of tubers.
Yeah, it's more advantages, more number of tubers, and again optimized, like, what should be the size of the tubers to be harvested. So once we optimize that, like, we did an experiment. I put one of the students, MSc students, to optimize this the tuberization, I mean the tuber size, and we found that about 4 grams of the tubers is the one which is needs to be harvested. For example, if you don't harvest that and leave it as it is, it will be grown bigger. That is not the one. That's baby tubers. They are baby tubers. The baby tubers are slightly bigger. No. It's a baby, like we have different kinds of tubers that you get. That the tuber that we get in market, which you consume, is a bigger one. We do get what is called as small tubers. We call them as baby tubers.
That is different variety.
No, that, like, there are different varieties are within the, like, when you grow them, there are some cases like, the tubers are sizes minimal, like, when they make a gradation, they will get the baby tubers as well. So what I'm talking about is the mini tubers. That we also another concept called the micro tubers. I'm not talking about that. Okay. So these mini tubers, like, the advantage of this is, as and when these tubers are being harvested, there is more and more option for the production of the tubers. That. So that is one of the greatest advantages of the growing the potato plants in aeroponic chambers, because as and when you harvest that, you can produce the more and more tubers. That. So that way, unlike the seed grown plant, I mean the soil grown plants, so where the harvesting is possible only in the end, but here in this case, as and when the tubers reach to a size of about 4 grams, you can harvest them and allow these plants to produce as many as possible. And this work we published in 2002 in Environmental Experimental Botany. This is what, like, I was mentioning about the importance of the temperature, like how the temperature is really playing a role as far as the tuberation is concerned. That, like, if you know what we have done was, as I told you, that we could regulate the root zone temperature. However, the shoot zone temperature was not been possible to be regulated. But although we now regulated the shoot zone temperature now of late, but initially we could regulate only the root zone temperature, not the shoot zone temperature. So in order to see what is what are the effects of the altered temperature or increased temperature on tuberation, what we did was we took up the planting at a different the time interval. The first planting was taken up at the right time, that is the Rabi season, sometime around in the month of the October, where the Rabi season starts. So then we took up the planting again in the second, the middle of or later part of the November, and the third planting was taken up in the month of the December. Now the idea is, because as you go, as you go away from the Rabi season, the temperature keeps building up, and therefore we wanted to examine what will be the effect of the increased temperature on tuberation. So we have done, we have seen that, for example, in this case, the first planting, the shoot zone temperature was 22.2 degrees centigrade to 26.9 degrees centigrade. That was the range. And the second planting, the temperature range up to 28.2 degrees centigrade. And more importantly, that if you see that the temperature, the shoot zone temperature was increased about to 32 degrees centigrade in the third planting, when the planting was taken up in the month of December. However, we could maintain the root zone temperature at around 19 degrees centigrade, 19 degrees centigrade plus or minus 2 degrees centigrade, and the non-regulated root zone temperature was 23 to 24 in the first planting, and in the second planting it was 24.6 to 26.9, and in the third planting the root zone temperature was increased from 26.9 to about 27.3 degrees centigrade. So that means if you delay the planting, both the root zone as well as the shoot zone will be exposed to the higher temperature. And we know that higher temperature is not favoring the tuberization. Only when you take up the planting and the right condition, right time, that is Rabi season, you could see that the tuberation. So that's what we got. Now you see that the mean cumulative temperature, and as you see that the mean cumulative temperature keep increasing, especially like the day temperature, the shoot zone temperature. You see that the shoot zone temperature is keep increasing from first planting to the third planting. Similarly, like when the root zone temperature, when it's not maintaining, the root zone temperature also keeps increasing. But however, if you now look at the root zone temperature is rather been constantly maintained, so suggesting that if you could maintain the root zone temperature through the spray of the chilled nutrient solution, it's possible that the temperature is to be maintained. Now look at this data, like when the in the first planting, when the root zone is rather being, for example, when the root zone being is maintained the temperature of around 19 degrees centigrade, you could see that the tuberation, more tubers are being produced. That whereas when the root zone temperature is not been able to be maintained, the tuberation is poor. That. But look at that the temperature, like even with the see in all the second planting and the third planting, although there is reduction in the tuber number as compared to the first planting, because remember the shoot zone temperature keeps on increasing. That if you delay the planting, the shoot zone temperature keeps increasing, and that's also affecting the tuber number. But however, through the regulation of the root zone temperature, we could minimize the, or rather minimize the damage caused by the increased or elevated temperature. And more so that hardly will see any kind of the tuberation in the third planting, where the planting was taken up sometime in the December, mainly because of the fact that the temperatures are higher, like especially the temperature coinciding with the tuberation period was higher, where the shoot zone temperature was 32 degrees centigrade, and even in the root zone also it was around 27 degrees centigrade. So therefore, these conditions are not really favoring, and hence the tuberation was very the tuberation was not there in the delayed planting. That it suggests that the potato requires an ideal conditions of the low temperature which prevails only when you take up at the right time of sometime in the month of October. So this is the data that we got it, like as you see that from the first planting to the third planting. So the first planting, the tuber number was higher with the root the temperature regulations, where the tuber number was almost close to about 30 tubers produced when you when you take up the planting sometime in the October. And as you see that in spite of the root zone being regulated, the tuber number keep increasing when decreasing. That suggesting that if you delay the planting in spite of maintaining the root zone temperature, still you could see the reduction in the tuber number. It indicates that both the root and shoot, or the temperature of maintaining the root and shoot temperatures, are more critical for the tuberation. Okay, if only the root temperature is very critical, then one would have got the similar tuber number. That right. So since the tuber number keeps decreasing in spite of the root zone temperature being maintained at 19 degrees centigrade, so it suggests that the shoot zone temperature is also playing a very important role as far as the tuberation is concerned. And in fact, our recent data shows that where what we have done in the recent experiment, which I'm not showing now, since the student has not completed her masters. So what we have shown that if you could maintain both the shoot zone and root zone temperature, tuber number is definitely been increased, even in spite of whatever the planting time period is that. So suggesting that the both shoot and root zone are temperatures are very critical. And the title of the paper that we have given was again the same thing, like both the root and shoot temperature regulations is more critical for the tuberation. That is the reason that and in these cases the shoot zone temperature, for example, if you now look at it, like whenever the root zone, although the root zone was temperature was regulated, because of the shoot zone temperature keeps increasing, that it is affecting this thing. So the VPD also plays a very important role as far as the tuberation is inhibited. And even the tuber weight also follows the similar trend as that of the tuber number. And if you now summarize that, what we have done is that we have summarized that, for example, in this first case, the first planting, the first planting was sometime in the congenial conditions, where the tuber number was around 22, where the shoot zone temperature was 22 degrees centigrade, And when the shoot zone temperature was increased to 28 degrees centigrade, you see that there is a reduction in the tuber number. And in the third planting, when the temperature is still higher, where the shoot zone temperature was 32. And remember, in all these cases, the root zone temperature was maintained at around 19 degrees centigrade, suggesting that not only the root zone, but even the shoot zone temperature is also important. Now, what we also summarize the results of both the root zone temperature regulations and no regulations, and found that whenever you regulate the root zone temperature, you could still get higher number of the tubers compared to the non-regulated the conditions. So suggesting that either you can regulate the shoot zone temperature or the root zone temperature, and regulating both the root and shoot zone temperature, that will be in ideal conditions. That so if you have a provision of regulating the shoot zone temperature, you could still get some tubers. If you have a provision to regulate the root zone temperature, you still get some tubers. But if you don't have an option to regulate the temperature, you will not be able to get any tubers, suggesting that the temperature is playing a very critical role. Now, what is really responsible for the tuberation? How the tuberation is regulated, and the tuberation is regulated by what is called as the sort of a the flow mobile signal, where the signal is rather been moved in the form of the transcript. So where the signal in the form of SP6, that is the side pruning, the 6 that is the transcription factor, and the bell 5. So these are the master regulators which are serving as a mobile signal, flow mobile signal. They move from the shoot into the tuberizing zone, where upon moving, it is activating the tuber, activating a zone that, and therefore the tuberation is possible. That. So what I just want to tell you is, for these mobile signals to move from the shoot zone to the root zone or the tuberizing zone, it requires the ideal conditions of the temperature, cooler temperature, and the short wave conditions, which will facilitate the transcript or the phloem transcript to be moved from the leaf into the tuberizing zone, where they could induce the tuberation. So the tuber formation is rather been controlled by the homolog of the floral indicator, the flowering locus T, referred to as the SP6. So the SP6 produced in the leaves, transported to the tuber zone, initiate the tuberation, and that is responsible for the tuberation. Now, how this temperature regulates the tuberation at the molecular level? If you now look at it, there are two kinds of situations. What you could see is, whenever there is a high temperature, it is not favoring the SP6 and the bell 5, the mobile signals, to move from the shoot into the tuberizing zone. Whereas in the in the low temperatures there, where you could initiate the transcript to move from, because the signal has to carry it there in the tuberizing zone, and that's how the tuberation is possible with the temperature. And same thing also holds good for the other factors, like the short air conditions and all this. And mostly this kind of work has been done tremendous. So much of work has been done, not from our group, but some other people, where they showed very specifically how these the transcripts are moving, and helps in the tuberation of the potato. So in this case, SP6 and the bell 5 are acting as a positive regulators, whereas the bell 11 and SP5G are acting as a negative regulators. So these are the regulators which will either positively regulate or the negatively regulate the tuberation. So this suggests that the mobile signal has to move from the shoot into the root, tuberizing zone, for the tuber initiation in potato. So this is what the expression data that we have, the quantity. Yeah.
Previously, we told that SP6 is a homolog of the FLT. FLT will induce flower.
Yes.
In this case, it reduces tuberation.
Yeah, it's both. See, the difference is the same the SP6. It's a homolog for the flowering locus T, which is also inducing the tuberation. And in this case, the potato, the same the SP6 is induce the flowering when the conditions are the long day conditions. So the same fellow is doing both the jobs there. It is doing the job of serving as a mobile signal for the tuberation at the same time, and the short day conditions. Remember, under short day condition, it is doing the job of something like a herbicide, like 2,4-D. Right? When you use it in a low concentration, it is acting as a hormone. When it is using at high concentration, it is using as a herbicide. Okay.
What about you give temperature shoot and root, same temperature.
Yeah, that's what I told you. No, see, when you regulate this temperature, that's what we did recently. But that data I'm not showing for some reasons, but the student has not completed. So where we found that when you regulate both shoot and root zone temperature, the tuber number is still increased. So suggesting that, so if you have an option to regulate these two temperatures and both the root and the shoot zone, it is possible to maximize the tuberation further. So this is the expression analysis expression data for the SP6 and the BEL5. These are the two positive regulators where you can see where we analyze these we looked at the expression of these positive regulator as well as negative regulator. You see that when the shoot zone temperature was 22 like in ideal conditions, and you see that the expression of both SP6A and the BEL5 was higher, and as the temperature keeps decreasing, increasing, the level of expression of the positive regulators keep decreasing that. However, the negative regulators like SP5G, which is a negative regulator, as the temperature is increasing, the negative regulator is now taking an upper hand. Yeah, both leaf and stolon. Yeah, this side is leaf and the other is stolon. So I'm not I'm not sharing all the data because we have done the data with the other genes as well but I'm just giving you only a couple of those. Yeah, that's what all the signal will have to come from the shoot when the shoot system that.
Okay. Then what's the point of manipulating the temperature?
No. What happens is there is always a communication. There's always a communication that. So I told you that so nothing like maintaining the root and the shoot zone temperature because the conditions of the the signals that are moving is moving without having any kind of the problems that. Now that what happens is when the root zone temperature is being maintained that there is always a root and shoot communication that. Probably you must have heard that shoot probably Bhumi knows that root and shoot communication by Shashidhar. Okay, Dr. Shashidhar used to work on that, and there is always a root to shoot communication. I don't know, Babu knows about that. Yeah, so where we root to shoot communication will always be there. That see, it doesn't mean that the communication is not happening between the root and shoot system. That so the communication goes there. So when the communication is going from the root system into the shoot system, that there is some amount of the temperature is being maintained. That is that. So the level of the transcript that is moving is also depending upon the kind of communication that is going from the root into the shoot. Yeah, chiller. No. The root shoot temperature we could regulate we grew these plants in the polyols where the naturally ventilated polyols we could maintain the temperature that. And it was a natural ambient temperature in the Rabi season in the month of October the temperature was cooler that. Natural conditions. So with what we done was with the natural conditions with putting some layers of the shade net we could somehow bring down the temperature, right? So therefore the ideal condition at that time was initially in the first planting it was around 22 degrees centigrade. That was the temperature maintained, and all those things have been maintained with the different layers of the shade net that we use that. Okay, so we recorded the temperature on a daily basis. So that way we had enough regulated the temperature as much as possible without having any kind of the control in terms of using the chillers even to spray onto the shoot system. But recently we have modified the chamber and we see that the shoot zone temperature is also been possible to be maintained by regulating the temperature in the. Yeah, so what happens to Bhumi, the chiller temperature, the moment the chiller, the nutrient solution goes out of the temperature, the chiller was 15 degrees centigrade. So when it's now passing through the pipes, and by the time it goes into the chamber, so the temperature from 15 degrees centigrade has increased to 19 degrees centigrade. So that's what So by reducing the temperature, we made an attempt that, so that is not really working. So therefore, we thought that the ideal temperature would be with several experiments we have done that. Even we could reduce the temperature further, the result was not encouraging that. So therefore, it is unnecessarily waste of energy that. Say to bring down the temperature further, you need to incur a lot of energy that. So and without having any kind of advantage, there is no point in bringing down the temperature further. So ideally, we thought, we standardized that the temperature of 18 to 19 degree or even 20 degrees centigrade would be ideal for the root zone temperature. Jani, I think you have any questions? Okay, are you right? Any students? Any questions? Are you following, or you are out of the track?
Really new for
Is it, I mean, are you on the flock, right, on the right track? Yeah. Yes. It is a closed chamber. Yes.
But even then some of the light.
No. We have made it completely dark that. Only at the time of the harvesting of the tubers, we could open the tubers. Like, the chambers were made in such a way that 3 sides are completely blocked, like closed.
Almost to the root zone or?
Root zone, not to the shoot zone. We have created all those things, like, the darkness was completely created in the root zone, and only at the time of the harvesting of the tubers, we open the chamber and harvest that, which done in couple of minutes, maybe about 5-10 minutes. Otherwise, it used to be completely closed. So all those things we have been taken care of. Yeah. So we know that the SP6 and the BEL5 are the important of the transcripts which are serving as a mobile signal, which are necessary for the tubulations. And again, this is not our data, but I just pick it from the literature that whether the increased expression of the SP6 increases the tubulation, and found that you could see that the increased the expression of SP6 is indeed rather been useful in terms of increasing the tubular. If you look at this, we know that the short day condition is the one which is favoring the tubulations, and whenever you give the long day conditions, the tubulation is the expression is absolutely almost zero. That, but even for example, if you can overexpress this particular gene SP6A, even in long day conditions, you see that the tuber yield is you see that the tuber yield is increased, that suggesting that even if there is non-optimal conditions, if you somehow make this gene to express and serving as a mobile signal, then it is what is required is this signal has to go there, and it is inducing the tuberizing complex and make that ask the plant to produce the tubers. That sorry. Now somehow what is required is in natural conditions, like the temperature is the one which is now making the expression of this SP6A and BEL5 that. Now, with whatever the mechanism that you could possible to express this gene and see that the transcript goes into the tuber complex, and therefore the tuberation is possible. That, but under natural conditions, what happens is this mobility of this particular SP6 synthesis and mobility is dependent on the temperature and the photo. So, in another experiment, what they've done is they have overexpressed the SP6A codon optimization that forces the storage organ development, where you could see that the tuber number is increasing, in these the codon optimized the SP6 plants. You could see that the codon number, the tuber number is increasing, suggesting that if you could increase this SP6A, so it's possible that the tuberation is is possible, meaning that it's everything is being boiled down that whatever the conditions that I told you is ultimately boiled down to the kind of expression of these mobile signals which are now responsible for the tuberization. So you can also see that the tuber yield is increasing in those the overexpressed lines, suggesting that these are the important players for the tuberization. Okay. Now this is with respect to the temperature regulations and the mechanisms of how that temperature regulations is responsible for the tuberization. Now another important factor is the light. That now why we got this idea is that because when you grow these plants in the in the polyols and with a lot of layers of the shade net, we know that what will happen to the light, that so automatically the light will cut down, that depending upon the number of the layers of the shade net that you put that. So that is not really encouraging that the plant also need to we are not looking at only the tubers. Before the plants could produce the tubers, the plant has to put on the lot of growth. Only when there is vegetative growth, the significant amount of vegetative growth, then it could support the tuberization. So in this scenario, what we have done is we looked at what is the role of the tuber and the light on the tuber, and we found that again I'm not going into the details of it. What we did found that was the warm white light initially is rather been required for the effective the growth of the plants. That so where you can see that extending the light period in using the warm LED light during the pre-tuberation period from 21 to 45 days is helping the rather than the more vegetative growth. So you can see the difference between the vegetative, both of them have been planted at the same time, but this was not given the light, whereas this was given the warm white light from 21 days to 45 days. That is the active vegetative phase. And you could see a huge amount of vegetative growth of these plants. And that has turned into the tuber, like, you have so much of the resources in the, the plant, and that is helping the tuberous. And that has increased substantially the plant biomass. And later, what we did was we stopped the warm lighted because we do not want the further vegetative growth. Rather, we are interested in the tuberization. For the tuberization, the far red light is more important than. So here, what we have done was we have given after 45 days, that is the time at which the stolon and tuberization is forming. So at that point of time, we have given the far red light, which has induced the tuberization, and we found that with the warm light, we could get more amount of the tubers. So although I have a number of data, but I'm not sharing that at that point of time. So this is the data that I'm given, I would like to share with you. This is what is happening. These are the three different treatments. Tuber number with, don't say that is highlight, it is the amount of light that is normally required for the plant growth. So this is the one, as the light is given sufficient amount of light with the shoot zone temperature was about 22 degrees centigrade, which was prevailing in the first planting. And as the shoot zone temperature increasing, so you could also see that the tuber number decreasing in spite of giving the high light. But if you now look at this, the dotted one, this is the one which is then the tuber number with respect to the warm white light. If you give warm white light to these plants with different levels of the shoot zone temperature, so you see that when the shoot zone temperature, although the root zone temperature was maintained in all these cases, the root zone temperature was maintained at around 19 degrees centigrade all through that. The only variable was the shoot zone temperature at that point of time, because we did not have an option to reduce or regulate the shoot zone temperature. Yeah, that's what, for example, when you give this the far red light is shown to improve the tuberization. So over and above the whatever the far red light that the plant is getting under normal situation, if you are giving the far red light is not been given for a long period of time. That it was only for a short period of time. That, and again, remember that the warm light that we also given is not for a long period of time. That it's only for an extended period for about another 2-3 hours from the end of the day period. So likewise, in this case, the far red light was given only for about 2 to 3 hours. Okay, so it suggests that when you have the combination of the far red light, for example, warm white light and the far red light, you see that the tuber number is still increasing, suggesting that if you could increase the white light, which will favor the vegetative growth. Added to that, if you could provide the far red light, the tuber number or the tuberization is increasing, suggesting that not only the temperature, where we are not brought in the temperature, where the temperature of the root zone is being maintained, that, and when the temperature also been maintained in the root zone with planting at the right time, and providing the warm white light which facilitate the vegetative growth, and providing the shorter duration of the far red light, you could still increase the tuberization, suggesting that the tuberization is rather also been regulated by these the light manipulation. So this paper is under preparation, and maybe in a couple. I have been asking this guy to write the paper, and that the students are not taking the lead unless this is happening everywhere, right? Probably Johnny knows about that. So I have been pressing, it's been 1 year, Johnny. I've been telling him that guy is taking that. So it's under preparation. That. So how is that the tuberization occurs in a short day condition? If you now look at what really the kind of the mechanism that operates in the short day conditions, and how the short day condition is rather favoring the tuberization and not the long days. That hope I am on the track. All of you are on the track. Are you following? Okay. Now, so how the tuberization occurs under short day condition? So if you recall that BEL5 and SP6 are the positive regulators, whereas SP5G is a negative regulator. So these the transcripts should move from as a mobile signal. They move to the tuberizing zone, and therefore the tuberization has to happen. That. Now what happens is under short day conditions, which is favorable for the tuberization. So we have these the transcription factors called the gigantia and the flavin K-box, F-box protein. These are the two transcription factors So what happens is under short conditions, these two transcription factors, they remain separated without they binding to each other. So once they remain separately, so there is no the CDF, that is cyclin dock factor 1 is not being degraded. So when the cyclin dock factor is not degraded, then what happens is the cyclin dock factor is active, which will suppress the constants, and as a consequence of which the negative regulator like constant is regulating the negative regulators like SPC, SP5, and it is also directly affecting the BEL5 that. So as a consequence of which, since the CDF1 is active, that is now responsible for the tuberation. However, under long day conditions, what happens is these two transcription factors, gigantia and the flavin K FBOX protein, they bind together, and once they bind together, then it degrades the CDF1 that. If CDF1 is degraded, then what happens is constant will take an upper hand. It triggers the SP5G, which is a negative regulator. It also suppress the BEL5, and therefore you will not have an option for the BEL5 and SP6 to act as a positive regulator, and therefore there will not be any tuberization. Hope you understood that. Now this is with respect to, again, I'm sorry that I could not share much of the information because that data has not been published. That's the reason that I have restricted my talk only to 1 or 2 slides with respect to the tuberization, light-induced tuberization. We also looked at what will be the role of the hormones, the plant hormones. So I told you in the beginning, if you recall, that the GA is acting as a negative regulator, whereas the cytokinin is acting as a positive regulator of tuberization. So we know that the photoperiod, short day conditions, low temperatures are congenial for the stolonization as well as tuberization, and low GA and cytokinin is also important as far as the tuberization is concerned. And if you now look at this, whenever you give the BA, the cytokinin, and also the white light, warm white light, you could see that the tuberization is more in the wherever the root zone temperature is being regulated, that suggesting that the addition or the application of the cytokinin will rather be favoring the tuberization, and added to with that, if you could also include the warm white light, the tuberization is included. However, when the root zone is not being affected, regulated with high temperature, the tuberization is rather decreased, but it can elevate the tuberization to a certain extent when you give the BA and the warm white light. Yeah, the same thing is also holds for the tuber weight. So in this case, what they've done is to see what is the role of the gibberellic acid. They overexpress the gibberellin, and see that there is a delayed tuberization, and they also created the RNA lines for the GA20 oxidase. You know that the GA20 oxidase is involved in the biosynthesis, later part of the GA biosynthesis, and when you overexpress that, what will happen to the tuberization? When you overexpress the GA20 oxidase, that induces the GA biosynthesis, there will be more GA, and what will happen to the tuberization? Less tuber. So you see that the tuber number is less. You can see that here in this case, as compared to the control conditions, the transgenic plants where the overexpression of GA20 oxidase, the tuber yield is rather been reduced, that suggesting that GA is acting as a negative regulator. However, when you go for the RNA lines of GA20 oxidase, as compared to the control, you could see that the tuber yield is increasing, suggesting that if you have a mechanism of reducing the GA level, it's possible that you can increase the tuber yield. That paclobutrazol, I'm sure you have heard of paclobutrazol. What is the paclobutrazol? It's a G inhibitor. It's a G inhibitor. You can see that if you use a G inhibitor, you could see that the yield of potato is rather been increasing. So you can see that the different days of the application of the paclobutrazol, 28 days after planting, and you see that the yield is when you apply the paclobutrazol at the beginning, at the early stage, you could see that the yield of potato is rather been increasing, and as compared to the no paclobutrazol, suggesting that somehow if you could regulate the GA biosynthesis, GA, then it is possible that you can increase the tuber yield. This is again the starch content. You see that the starch content, even the quality has also been improved with lowering the GA at the time of tuberization. Again, remember the G has to be, see that the G has to be reduced at the time of tuberization, not at any development phases. Another one is the cytokinin, where you know that the cytokinin, if you can increase the cytokinin level, also you can see that the tuber number is increasing. That what they've done is they have they developed the transgenic plants over expressing the IPT gene, that is isopentyl transferase. That's an important gene in the cytokinin biosynthesis, where they could see that the tuber yield is increased with the overexpression of IPT, and also externally apply the benzyl adenine. So whenever they apply the benzyl adenine or they develop or they overexpress the IPT, so the tuber yield is increased compared to the control condition, suggesting that the cytokinin is rather been useful for increasing the tuberization. Similarly, the jasmonic acid has also been tried, where the development they use different levels of jasmonic acid, and they found that when they use the jasmonic acid as compared to the control condition, the tuber yield is increasing, suggesting that these hormones like cytokinin, the jasmonic acid, and the lower level of gibberellic acid, or even if you could control the GA level at the tuberization, it's possible to increase the tuber yield in potato. So all these reports suggest that the potato tuberization is rather been influenced by cytokinin, jasmonic acid, paclobutrazol, auxin. Of course, I did not use that the data. I did not show that. Even auxin also shown to promote the tuberization, but mainly the cytokinin and is the very key player which will influence the tuberization. However, the gibberellin is suppressing the tuberization in potato. Now, what we finally what we did was we brought in all the components. All the components means temperature regulations, light regulations, as well as the hormonal regulations, and see what will happen to the tuberization in potato. And in this scenario, what we have conducted was one experiment. This experiment was conducted in Kharif season. Remember, this experiment was conducted in Kharif season. Kharif season is not ideal conditions for tuberization, and we took a challenge like whether we could produce the tubers or not possible in Kharif season. And this is the evidence that it's possible even during Kharif season. If you could control many of the parameters of tuberization, it's possible to produce the tubers there. So where we grew the plants as usual, we grew the plants in aeroponic chambers. We regulated the temperature. We also regulated the red to far red ratios, where we maintained a lower ratio of red to far red as at the time of the tuberization. We also maintained. We also take up the spray of the cytokinin, and we also take up the spray of gibberellic acid, and also the paclobutrazol. And this is what the results that we got. So we used two different varieties. One is the Kufri Jyoti and the Kufri Malini. And you see that the data, when the temperature, in this case, when you use this, when you use only warm white light, this is what the data that we got it. So the black line represents where the root zone temperature was regulated, whereas the dotted line was representing the root zone temperature was not regulated. And you see that with the addition of this component, for example, when you give the far red light over and above the white light, when the shoot zone temperature was 25 degrees centigrade, the far red light has additionally improved the tuber. To this control conditions, if you add the PA, another treatment, the tuber is like in tuberization is increased further. And if you combine all those things, like combination of regulating the temperature, then providing the far red light, providing the hormone, and you could see that the tuber number is still increasing, that suggesting that through the combination of these the factors, it's possible to control the temperature. I mean, to control the tuberization or increase the tuberizations even in Kharif season. So to summarize this talk, I just wanted to summarize this talk. The tuberization is highly complex, regulated by many factors: temperature, photoperiod, hormonal conditions, and in this scenario, the SP6 and BEL5, they act as a positive regulators of the tuberization. All right. So this is what I just wanted to summarize with respect to the talk that I given. And we made some publications like environmental experimental botany. We published that, and as a compulsion, we also published in Mysore Journal of Agriculture. It's a compulsion. So no other options, because I don't know whether that compulsion is also there in TNAU that. So it's a mandatory. Whatever may be the data that is good, you need to put one paper into that, the Mysore Journal of Agriculture. This also another paper. It's a farmer innovation. It's not a very good paper, but we are likely to make at least another 2-3 papers from the data that we have generated in this the proposal. And one of my PhD students, Srikanth, we got some certificate of appreciation, where he presented and as one of the best startup awards, and also he got some cash prize for the work that he has done in in different places. And finally, I dedicate this entire thing to my guru, Dr. Uday Kumar. But for him, the department would not have been grown to that level, and but for him, probably I would not have been stand here and given a talk. That I am sure that the people from US Bangalore agree with me that he was a mentor for all of us, and I dedicate entire thing. I dedicate this talk and the work that. And incidentally, he was the PA of this project, and I was the co-PA of the project, and we were successfully completed, and it was highly appreciated by the funding agency. Thank you, one and all, for your patience.
From there, I say that TA is needed for a tuber Darwin's to break deep tuber Darwin's.
Darwin's is different. That even now that we know that tuber formation and tuber dormancy is different. That dormancy comes into the picture only when the tubers. See, the tuber dormancy is another issue in the potato, where we have the dormancy for about at least for about 2 months, 2 to 3 months. That is another big menace in case of the potato, where we people have not really today, even today, they have not come out with the dormancy breaking the protocol. So people try with the different combinations, like using GA, maybe using the temperatures and all those things, but it is not really any standard method is not available as of today to break the dormancy in potato. It all depends on the total surface area. For example, if the potato becomes bigger, you will have more hybrids. So in general, for example, in case of mini tubers, generally you'll come across only 1 or 2 hybrids. So if you leave these mini tubers to grow bigger, there will be more hybrids being formed, like what you get it in normal tubers that you consume.
Good afternoon, sir.
Madam, somebody is asking questions. I'll get back to you. Hello.
Yes, sir. Good afternoon,
Yeah.
sir. Hi, Mohan Raj, sir. I hope you remember me. See, I joined in 2017 batch PhD, Vani. I hope you remember me. Bharat batchmate.
Vani, your student. Actually, she joined.
Oh, yeah. Tell me, Vani.
Bharat, hi, sir. How are you?
Yeah, fine, Vani. How are you?
See, I'm good, sir. First of all, I'm very my heartiest congratulations to you and Srikanth to got that award, startup award, and you are doing a tremendous job in potato. So I'm very happy to seeing again and again our greenhouse area, everything I seen in photo. I'm very excited. So yes, you said we are missing a lot, Uday Kumar sir. So without him, so that much difference in our department in GKVK is not happened.
Right. Yeah.
Sir, about coming to subject, my question is about. Sir, is there any water stress related? Research you are doing in tuberization?
No, we have not done any work with respect to the drought stress on potato, because the moment you induce the drought stress without conducting experiment, I can tell you that openly that it will affect the tuberization, because people who grow potato, who cultivate the potato, will have to ensure that the moisture content is always be maintained higher, so that the proper tuberization is happening. Okay, so that's the one crucial thing. And tuber, the temperature and the moisture stress, they are the two crucial things.
Yes, sir. One more thing,
please.
sir. I want to know from you. From your experience in research towards potato, so tell me the interesting physiological story about brown core or halo hot.
Sorry. Can you repeat that?
is there any interesting physiological story related to brown core disorder in potato?
She's around. Maybe I can talk to you later. Like, you know, I mean, your question was not like, you know, I couldn't get your question.
Sure, sir.
I'll
Thank
get
you,
back
sir.
to you. Surely I'll get back to
Yeah.
you.
Thank you, sir. Thank you. Bye, sir.
Yeah, Lokesh.
Then only condition.
Yeah.
We are already discussed, say,
If you're not expressing, like, it's not apply. It's you're increasing the expression of that. Yeah.
whether it's temperature.
Yeah, it is technically. See, all those, like, you know, gene expressions, for example, a gene expression, like, you know, if you now look at the gene expression, probably Johnny agrees with me that, like, you know, when you give the stress, like, you know, you can see the whole set of genes, the set of genes are expressed. And when you, like, you know, give the stress at a different developmental stages, some other set of genes get upregulated, some are downregulated. So what happens is, for this expression of these positive regulators, which will induce the tuberation, requires the positive, like, you know, the ideal conditions in terms of the Shorter conditions and the temperature, which are acting as a signal for the expression of these the regulators, positive regulators. That's how everything, like, you know, as a stimulus. We require light as a stimulus or the temperature as a stimulus for the downstream events to take place. Okay. Any other question? That's all. Yeah, please. Yeah. Low nitrogen at the time of tuberization. No. I told you, I think I probably missed that. The nitrogen is required for the vegetative growth, but when it comes to the tuberization time, you'll have to, like, you know, see that the nitrogen level is reduced. I think Johnny had some question, like, you know.
Sir, this I have seen it.
Okay.
I'm just imagining how the
Okay.
just after seeing the photos.
Yeah.
I just saw that it has a very powerful system. It's just stolen.
Yeah.
From that gravity.
Okay.
Tubes. So a stolen comes from a place, but it goes electronically.
Yes.
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 So it's just like the cytodome or wheat. So it grows at point where. So when in a storage is a place where both root and shoot will also
No. Stolen is a modified stem.
modified.
It's a modified stem. Yes. Yeah. What happens is initially stolen comes, and after that it starts swelling. That's how the tuber is being formed.
So then what a stolen means a single root.
Yeah.
All the tubers will be in that.
Yeah, no. It's like, you know, all the roots, like, you know, yes, let us say root system. The stolen is independent of the root system. Independent. So it's a stolen is coming from it's a modified stem. So
there will be so many.
yeah, so many stolons are initially being formed that, and after that, like, initially elongation is taking place, and subsequently the swelling of these stolons, and that's how you will get the tubers. So bigger size tube.
Similarly, there will be so many.
Yes. Something like in a groundnut. You've seen the groundnut, right? Root system is there. Like, you know, you have the root system independent of the stolon. Yes. And this root system, I also forgot to tell you that when you grow these plants in aeroponic system, the root system will be totally different than from what you get it in the land grown plants, because there are no hindrance in the aeroponic system. So therefore, you will tend to get more of the finer roots compared to the kind of the taproot system that you find in the land grown plants. So all together different the root architecture you could see in the aeroponically grown plants. I want the questions from the students. Although the Lokesh is a student, not like, you know, I want from the girls, like, you know, the white the shade nets. No, we did not use different colors. Probably there could be a possibility, like, if you use different colors, that will affect. Yeah, those things are there. But yeah, but we used only the white the nets, shade nets. Which one? Tuberin. We did not look into that. I don't know much about that. Yeah, everything is regulated that. For example. Yeah. So what we did was, like I showed you in one of the slides, that we have standardized the nutrient solutions. We also standardized the on and off system, that, you know, the motor has to be switched on for some period of time, spray the nutrient solutions, and keep silent for next 10 minutes. And likewise, every 10 minutes it used to switch it on. and the duration is again altered during the night time. So we have programmed, everything was programmed. So it was not manually done that, everything was controlled. Hoagland solutions. And another thing is I did not share many of the things that we standardize the Hoagland solutions. We have taken those number of the nutrient solutions that have been recommended. And finally, we standardized our own thing where we included or increased the nitrogen and the potassium and also the calcium. I think it answers the location questions. So initially, we increased the nitrogen content initially to boost up the vegetative growth. And finally, when we start giving, like when the plant has entered into the tuberization time, at that time, we have brought down the nitrogen level and see that the nitrogen level is being coming down.
What's the pH of that field?
The pH was around neutral, like around 6.5. That was the.
Thank you, sir. It is Eman Professor for giving a vote of thanks. So because of all busy schedule, you are coming over here accepting for Viva and guest lecture. Okay, so it's a very good, excellent lecture, sir. All the students may be benefited. It's also new to us, tuberization potato. So once again, because of our department, so we are giving a very good thanks for giving a good lecture. Thank you, sir.
Thank you.